Today we will discuss Chapter 9. Previously, in the first 5 chapters, we discussed the introduction of the biochemistry. Also, we discussed amino acid, peptides, and proteins, protein structure, and also the technique to purify protein to characterize the protein properties. And starting from chapter 9 until chapter 13, we basically will discuss nucleic acid, including the structure of the nucleic acid, and also those cellular activities that involve the nucleic acid, including transcription, translation. So that also include the protein synthesis. And in chapter 13, we will also discuss the nucleic acid biotechnology techniques. And so all these are based on the nucleic acid from chapter 9 through 13. So in chapter 9, basically we will discuss the structure of the nucleic acid. And in this chapter, we will have several topics to discuss, including the level of structure in nucleic acid, the covalent structure of polynucleotide, the structure of DNA, denaturation of DNA, and also the structure of the RNA. And in chapter 4, remember in chapter 4, we identify 4 levels of structure for the protein, including the primary structure, secondary structure, tertiary structure, and the quaternary structure in protein. And nucleic acid can be viewed in the same way. The primary structure of nucleic acid is the order of bases in the polynucleotide sequence. And the secondary structure is the 3-dimensional conformation of the backbone. And finally, the tertiary structure is specifically the supercoiling of the molecule. And there are two principal types of nucleic acid, as we all know: DNA, deoxyribonucleic acid, and RNA ribonucleic acid. And so this chapter, basically, we will discuss all these structure.
The transfer, there are 3 different pathways, okay? One is the transformation, which we will discuss right in the next few slides about transformation. That's how the bacteria cell perform this horizontal gene transfer. And number 2 will be the conjugation. Number 3 will be the transduction. So the transformation, basically, that will be you incubate the bacteria cell, all right, with the DNA. And then the bacteria cell will uptake the DNA. That's called transformation, which we will explain in the next few slides. So that's the first mechanism for the bacteria to exchange the genetic material. And the second mechanism, the conjugation. So that means one cell with the pair and the other one does not have pairs. And then with the one with the pairs, that's the F plus bacteria, they will find the F minus. All right, because the F plus, they have this F plasmid. So they will try to bring the F minus cell to the close proximity, and then they can transfer the DNA, the F plasmid, to the F minus cell. So the F minus cell become F plus. Okay, that's conjugation. And then transduction. Transduction, that will be that will go through the virus. Okay, that will be the bacteriophage. The virus, they infect the bacteria A, and then they perform those propagation, and then lyse the bacteria A. And when they package the genome, they accidentally also package the bacteria A chromosomal DNA into the virus. Then they lyse the bacteria A and infect the bacteria B. And during this time, the bacteria A chromosomal DNA will be transferred into the bacteria B. So that's called transduction. Okay, so these are 3 major mechanisms for the bacteria cell to perform this gene transfer. Okay, and as I say, the first mechanism will be the transformation. So transformation, basically, that's the first experiment to demonstrate that there's a genetic material can be transferred. Okay, can be transferred from one bacteria cell to another bacteria cell. Okay, that will be in the 1928, almost 100 years ago, performed by this Griffiths, very famous experiment, Griffiths experiment. So basically, he observed the change of the Streptococcus pneumonia cell from the virulent, okay, the rough colony bacteria without capsule, to the virulent, the smooth colony bacteria that has capsule. All right, so the virulent, the S cell. Okay, and then the result is he killed the virulent bacteria, can transform the virulent bacteria into virulent bacteria. Okay, the experiment is like this. So he performed several experiments. The first experiment, he inject this live S cell, all right? S cell, this virulent smooth cell, okay? And then the mouse die, all right? And then if he inject the live R cell, which is a virulent cell, then the mouse leave. And if he use the heat to kill the S cell, all right? This S cell, and then inject to the mouse leave, all right, because all the cell die. However, if you use the heat to kill the S cell, and then you mix this material with live R cell, all right, after the incubation, then you inject to the mouse die, all right. So that suggests something from the S cell being transferred to R and convert R to S so that they can kill the mouse. All right, and this process that later on we call it transformation. Okay, this is what he observed, and then later on people try to identify what is the genetic material in the S cell that can be transferred to the R cell to convert R to S to kill the mouse. Okay, so that'll be later on, like in the 50s. These three scientists, they perform this experiment. Okay, so again they collect the material from the S cell, all right, and then they collect it, and then they use different treatment, okay, to inactivate the different part of the molecule in the cell, and then they inject to the mice, and then to see what happens. So, for example, if they want to determine whether the protein is a genetic material, then they collect the S cell, all right, and then they treat it with protease. So that means the protease will degrade all the protein, and then they inject into the mice. If the protein is a genetic material, then they inject to the mice, since you use a protease to degrade all the protein, all right. So then the mice will live, will be healthy, all right. However, if they treat with the protease and then they inject to the mice, they observe the mice still die. So that means the genetic material is not the protein. And then they treat it with the RNase, which is the enzyme that would degrade RNA. And after that, they inject into the mice, and the mice still die. So that means the RNA is not a genetic material. And then later on, they use the DNase, all right, which is the enzyme would degrade the DNA. After they treat the material with the DNase and then inject to the mice, then mice live. So that suggests the genetic material is the DNA. So this is the first experiment to demonstrate that DNA is the genetic material. Okay. And then also in 1952, the two scientists, Hershey and Chase, they demonstrate that the virus, okay, the bacteriophage, also use the DNA as a genetic material. Okay, so what they did is they basically they grow the bacteriophage. Okay, they let the bacteriophage to infect the bacteria cell that has been grown in a specific media. All right. So, for example, the media that contain P33, which is the isotope for the P32, And later on, when we learn the DNA structure, we would know the DNA carry the P32, all right? So they use the P32 to feed the bacteria, and then let the bacteriophage to infect the bacteria. And so that if the genetic material is the DNA, then the bacteriophage will carry the P32. Okay, and also they in another parallel experiment, they let the cell, all right, bacteria cell grow in the presence of S35, right? S35 is the isotope of the S30, the S32, all right, and then the S35, okay, basically is isotope, and then if the genetic material is protein, then the virus later on they will carry the S35. So based on this experiment, and then they identify the P32, the P32 is inside the virus, and so they confirm that the genetic material is DNA for the bacteriophage. All right, and then another example for the virus is this one. So as you all know, that the genetic material for this virus, the COVID-19 SARS-CoV-2, is a positive strand RNA. So that shows you the RNA can be also the genetic material. Okay, and later on we will have a section to discuss the SARS-CoV-2. Okay, and then also the DNA is the genetic material for the eukaryotic cell. Okay, so this can be proved by the genetic engineering. So we can purify the DNA and then clone into a specific vector, and then we can express the specific gene, and then to get the product. Okay, so from the genetic engineering that we know that DNA is the eukaryotic genetic material. Okay, that's the first section about to prove that nucleic acid is the genetic material. Okay. Now the next section we will discuss the structure of the DNA and RNA. Okay. So the DNA is a polymer, right? Polymer, that means each one is a monomer. Okay, each nucleic acid is a monomer. Then you can form the polymer through the dehydration reaction. Okay, and here I show you the 4 different structure. Okay, we have the pyrimidine. All right, so these 2 are pyrimidine. Okay, we have the thymine and cytosine. And purine, we have this adenine and guanine. And the reason we call purine pyrimidine because the structural similarity. So we call it pyrimidine and purine. And pyrimidine, this ring, similar to the pyrimidine, and purine ring here, adenine guanine. And the nucleotide, basically nucleotide, okay, nucleotide basically is a phosphorylated nucleoside. So nucleoside, we have nitrogenous base, sugar, and then if this one is a phosphorylated, so that means you add a phosphor group here, that become the nucleotide. And this is the reason why here we have this P32, all right, the P32, all right, because this phosphor group here. And these 3 components that give you the nucleotide. Yes, Marina.
When you add that phosphate, is that the phosphate diester linkage bond, if I remember correct?
Yes, we will discuss that. Here will be the phosphor monoester bond, all right? Between this carbon and this phosphor group, this is the phosphor monoester bond. And only here, from this 3' hydroxy, connect to the next phosphate, right? That would be the phosphodiester bond will be here, okay?
Okay. And the purine, what makes it physically different from a pyrimidine? That it's just a double ring structure?
Yeah, double ring, that's purine, and one
and
ring.
And then pyrimidine is only one ring structure.
Correct, yeah.
Okay.
And here, just to show you how they form this monomer, okay, through the dehydration reaction. So, for example, glycosidic bond between the nitrogen base and the sugar. And this is the number of the carbon, okay? So 1, 2, 3, 4, 5, all right? And from this, and here it says 2' deoxyribose. I mean, this number 2 carbon here, see that deoxide, all right? Deoxide, it is hydrogen instead of the hydroxide. So it's deoxide. So that's why we call this deoxyribose. So that's why it's called DNA. And this is glycosidic bond, so through dehydration. And here is a phosphomonoester bond, the dehydration, all right? And as I mentioned, here, when you form a polymer, right, then you will have the phosphodiester bond, right, from this 3' hydroxy group to connect to the next nucleotide. And that gives you the phosphodiester bond. And here's a phosphomonoester bond. And this is just to show the phosphorylation, okay? Phosphorylation. And so from here, this is adenosine monophosphate. So this one we call alpha. When you add another phosphate group, this is called beta. So if there's 3, the triphosphate, all right? So alpha, beta, and gamma. That's how they name this triphosphate, all right? And then difference between DNA and RNA, all right? The major difference here, the 2' deoxyribose, 2' ribose. Here you have oxygen. Here you have no oxygen, okay? And then also the DNA, you have this thymine and have been replaced by uracil. And the difference here, you see this hydroxy group, and this is a methyl group, all right? The methyl group has been replaced by the hydrogen, all right? That change from thymine to the uracil. And then this is the polymer, how they form the polymer, okay? So basically, originally you have this, the nucleotide, all right? 5' because this is 5' carbon, all right? 5' carbon, yeah, yes? Yes, Mama.
Yeah.
Yes, what's your question?
Can you go back for the last? Yes, not this one. Yes, this one. No,
this
no.
One.
No, this one. This one.
Yes.
When you count the uracil RNA, you count from 1 or from nitrogen, Professor?
Nitrogen?
Yeah.
They come from here. See?
No. I mean,
they count the carbon.
From the carbon, not from the nitrogen.
Yeah, they come from here. 1, 2, 3, 4, 5.
Because
this one they count. Yes.
Because this one is counted from nitrogen, this one is counted from carbon, right?
Yeah.
Oh, okay. Thank you, Professor.
You're welcome.
Is that also another difference between the DNA and RNA, the way it's counted?
Yes, that's a small difference, but the major difference is here, all right? Major difference is here.
What creates that small difference?
The difference here, oh, it's a big difference.
No, I'm talking about when I'm counting the carbons, how it starts on the first nitrogen versus on RNA, it starts on the hydrogen.
Oh, you mean why this is different?
Yeah, like the physical counting, like why is that different?
That's a great question. Maybe you can ask the chemistry professor. Right? Did you take the Ogo one?
Yes.
Okay, so next time you can ask the Ogo professor. Then the Ogo professor will explain this in great detail for you. All right?
Okay, Professor, thank you.
So we focus on biology part, okay? Yeah. And then, okay, so now here. So this is the first nucleotide, okay? If you can see. Can you see my pointer?
Yes.
Okay, good. So you see, this is the first nucleotide. So when you want to form the polymer, all right, you have to add the next nucleotide, okay, from this three prime carbon, right? And then you connect this phosphate group to the 3' carbon. Originally, you have a hydroxy group, and now after you connect to this oxygen, all right, you will remove a hydroxy group, okay? That's a dehydration reaction. And then when you connect this to here, all right, so now this phosphate group, you connect one to this 3' carbon, and then you connect to the 5' carbon. So that's why we call this place here with the phosphodiester bond, right? We have this 1 and 2 phosphodiester bond. Okay, this is called phosphodiester bond. And then you can continue to form, to add the nucleotide to the 3' end. And then here I say 3' to 5' phosphodiester linkage because it's coming from 3 to 5 here. But overall, it's from 5' end to the 3' end here. Okay? Okay, that's why we say the polarity is 5' to 3', the DNA 5' to 3'. And then from this diagram, okay, you can see this side is always the same. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. This is the same. The difference is only inside the nitrogenous base. Okay? So that means this can be replaced by anything, but this sugar phosphate base is always the same. Okay? And this is just to show the RNA chain. Okay? And then from here the double helix. So because the hydrogen bond, okay, between the AT, we have 2 hydrogen bond, and GC 3 hydrogen bond. And so you can form the double helix. All right. And then this is 5' to 3', and then the opposite side 5' to 3'. Okay. So we call this is anti-parallel. Anti-parallel. And then outside sugar phosphate backbone are the same. Inside nitrogen base are different. And because they form a hydrogen bond, so they can stabilize the double helix. And so basically the double helix DNA that just meet this requirement for Chargaff rule, all right? So the 2 DNA strand, anti-parallel, so 5' to 3' orientation run in opposite direction. And each turn is 34 angstrom, which is 3.4 nanometer. So that means each nucleotide, that each nucleotide, each base pair, right? We call this a base pair is a 3.4 angstrom, okay? 3.4 angstrom. So 10 base pairs, 34 angstrom long, and the diameter is 20 angstrom. So that's 2 nanometer. So 2 nanometer, that's here to here, right? That's called diameter. And then the sum of the purine equal to the sum of pyrimidine, the percentage, so the amount of the adenine always proportional to the amount of thymine. And then the G proportional to the C. So that means if you have a 10% of A in the genome, then you will need to have a 10% of T in the genome. And then also, because you have 10% A, 10% T, so that you have 40% G and 40% C, all right? If you have 20% A, then you will have 20% T. Then you will have 30% of G and 30% of C. Okay? That's how you calculate the amount of the A, T, G, C in the genome. And then because 1 full turn, all right, 1 full turn, 10 nucleotide, 10 base pair, and 34 angstrom, then that means each base pair will be 3.4 angstrom, and 1 full turn is 10 base pair. So that means each pair, all right, from the first base pair to the next base pair, they have to rotate 36 degrees because 1 full turn is 360 degrees. So for to have the one full turn of double helix DNA, 10 base pair, 360 degrees. So each base pair has to rotate 36 degrees. Yes, Marina.
Can you repeat what you said about the base turning? You said every 10 base pairs is a turn?
Yeah, every 10 base pair is one full turn. And then, as you know, one full turn is 360 degrees. So that
Okay.
means each base pair from neighboring base pair, they have to turn 36 degrees.
Okay, I see what you're talking about now. Okay.
Yeah, sure.
And what did you say about the unit? It was on the picture, 12A. What does that stand for again? Where it has 12A and 22A, and then it has nanometers underneath. What does the A stand for?
Oh, that's Armstrong. Armstrong.
What unit
You know,
does that represent? Or is it just a unit
that's the
itself?
name of the unit. That's the name of a unit called Armstrong.
Okay.
It's a Armstrong, Armstrong, Armstrong.
It sounds new. I don't think I've ever heard of it.
Armstrong. So basically, every 10 Armstrong will be 1 nanometer. And then
Okay, so it's extremely, extremely tiny.
correct. Yeah, very tiny. Okay, that's called Armstrong. You see A, and then on top of the A, there's a small circle. That's called Armstrong.
Okay.
Good. And then the nucleic acid, they can hybrid by base pairing, right? As we mentioned, ATGC, they form the hydrogen bond, so they can form a double helix. And later on, when we discuss some molecular technique, it is very important to understand this concept, the hybridization, because hybridization, I mean two nucleic acid strand, single strand, when you mix together, they will hybridize because they will form this hydrogen bond automatically between AT and GC. If they are 100% complementary to each other, they will form a strong hybrid. Okay, and if it's less perfect match, then although they can form the hybrid, but they are not as stable as the 100% complementary. We call it complementary sequence. So that means A always match with T, G match with C. That's complementary, okay? And so the nucleic acid, so DNA can form the hybrid with DNA, and the single-strand DNA can form the hybrid with RNA, and then the RNA can form the hybrid with RNA, okay? And because RNA, they can form this double-stranded region, so RNA can have several different kinds of secondary structure, which we will also discuss later. And physical chemistry of nucleic acid. All right, so basically DNA, RNA, they can appear in several different structural variants that we will discuss, especially the DNA based on the relative humidity. Then the DNA can be the right-handed or left-handed. Okay, so this is the rule to explain the right-handed. So basically, 5 prime, okay, go from the right side to the bottom. All right, so this is called right-handed. If it's go from this, right, if this one in the front, 5 prime to 3 prime, it's go from left to right, so it's left-handed. So remember, this is right-handed, this is left-handed. All right, 3 different type of DNA, and we commonly seen is a bDNA. All right, so it's right-handed. So you can see this pink one from right side to the left. Okay, from right side to the left. Okay, and right side to the left, the pink is in front. Okay, so it's a right-handed. Right-handed DNA is it present at the physiological condition. All right, and 10 base pair per turn, but more specific is like a 10.2, 10.5. All right, but normally we refer this as a 10 base pair per turn. And then another right-handed DNA, it's aDNA. aDNA also right-handed, and only exists in the low humidity condition. Okay, in the normally in the cell, more than 90% is bDNA and less than 10% is aDNA. And also another one is called a zDNA. It's left-handed DNA. Okay, it's a extremely very tight. So 12 base pair per turn. Okay, so it's very tight. Very tight, fully it's a tightly packed DNA, and present in the very high concentration of the positive charged ion. So like a sodium or the magnesium. Yes, Muhammad.
What does it mean that the aDNA has low humidity?
Low humidity. So normally the humidity like 80%, 90%, then you have the zBDNA. Low humidity, that dehydrated.
Refers to oxygen? H2O?
No, yeah, H2O.
Okay.
Okay, dehydrated condition.
Okay.
Dehydrated condition, then the bDNA will turn to aDNA.
Okay. Thank you.
Yeah, welcome. And then another important property is, another, Marina.
Wait, it's referring to that the last slide. So the physiological conditions are going to basically influence what type of strand of
The
DNA.
DNA physical structure, exactly. The physical
Okay.
condition
So
will
I mean, this can just be in a general sense. So the bDNA, for example, is probably most eukaryote, prokaryote cells that we're
correct.
looking at.
Yes,
Okay.
it should present everywhere in the cell in
And
normal
for
circumstances.
aDNA,
Normal aDNA
sorry.
is very small percentage inside the cell at the place where there might be less water environment.
Would those be like
Okay.
extremophile microbes, something along those lines?
Yes, some yes, exactly. And remember this hypotonic condition.
Yes.
Hypotonic condition, that DNA can turn into aDNA.
Okay. So the aDNA basically condenses because there's less water.
Yeah,
so it
and
kind of tries to
zDNA
protect itself more.
Again?
Because there's less water, it basically condenses and tries to, I guess, turn in more and protect itself.
Yeah, and then the zDNA even more dehydrated condition, because you have very high concentration of the positive charged ion, and then they pack even further, so you have 12 base pair per turn.
Thank you.
That's zDNA, and then some other physical property. Okay, the DNA denaturation. All right, DNA denaturation and the secondary structure of DNA, okay, and tertiary structure of DNA. For the DNA denaturation, as I mentioned, the DNA, if you have the complementary sequence, they can form the double helix automatically, all right? And then the DNA denaturation, when you increase the temperature, so for example, this is the curve you can see here. If you increase the temperature from 70 degree to 100 degree, okay, the x-axis, that's the temperature. Increase the temperature, and then this is a percentage of the population of a single-stranded DNA and double-stranded DNA in the percentage of the single-strand, double-strand DNA in the population, all right? So, so as you can see, almost 100% would be double-stranded DNA in the low temperature, in our normal temperature, okay, that's 30 degree, all right? When you increase the temperature, all right, when you reach to a temperature where you can see 50% of the population is double-stranded, 50% is single-stranded, then we call that temperature is the melting temperature, that's Tm. And then if you continue to increase the temperature, eventually 100% of the population will be single-stranded, okay? because the temperature, they can break the hydrogen bond. So those ATGC complementary sequence, they would be pulled apart, okay? So that's the melting temperature, DNA denaturation, which is a very important concept for all the molecular technique that we will discuss in the future during the semester. And then there's secondary structure, so and the tertiary structure, so DNA. So as you know, for example, in the bacteria cell, those DNA, okay, are the circular DNA, okay, and circular DNA, and also we have some engineered plasmid DNA, okay. The plasmid DNA also circular DNA. Circular DNA, as you can see here, that's a circular. So these are double helix circular DNA, all right? However, they don't exist like this, okay, in the cell. They exist like this, supercoil, all right? Supercoil. So that would be the secondary structure, tertiary structure, right? And then when those DNA, okay, turn to this structure, then that come to another concept. It's called topology. Okay, the topology basically, so basically the topology of the DNA that can be described as the linking number, right? Linking number, okay, LK, let's call linking number. Basically, it will be the total of the twisted number and the writing number, okay? The twisting number plus the writing number give you the linking number. So what is the twisting number? So twisting number basically that will be the twisting number would be the turn, how many base pair per turn, right? That will be twisting number. So like in this case here, this example, so we have 210 base pair, all right? 210 base pair. If you consider the condition is 10.5 base pair per turn, then you divide the 210 by 10.5. All right, 210 divided by 10.5, that gives you 20. So that would be the twisting number. Twisting number. Okay. Twisting number, that would be this how many turns. And then the writing number. Writing number, basically you twist this double helix circular DNA. All right. Every time you twist one time, then that'll be one writing number. Depend the right turn or right-handed or left-handed. And in this case, this is right-handed, okay? Right-handed. If you turn this to the vertical condition, you will see this is right-handed. So the writing number is a minus. So it's a minus 1, 2, 3, 4. So the writing number is minus 4. And then the linking number will be 20 minus 4 is 16. Another situation is if you relax the DNA, okay? You relax the DNA, you open up the double helix for 4 turns. Then the linking number minus 4, that'll be 16. Okay, so this is a concept that the reason we discuss this is later on when we discuss the replication, transcription, translation, it's all relevant because during the transcription and the replication, when you add the protein to the DNA, that will increase some, that will change the DNA physical property, so that linking number will change. Especially if, so for example, during the replication, that normally that the DNA they have negative charge, negative supercoil, but when you perform the replication, they will introduce a positive supercoil ahead of the replication fork, all right? That will increase the tension, all right? And then we will need an enzyme to release the stress. Otherwise, they will break the DNA. So that's why we need to discuss this briefly, okay? And later on, we'll show you a video, which is very important to explain this concept in great detail, okay? So So supercoil occur in the closed DNA with no free end. That's in the live cell that you will see. So this is linear DNA. This is relaxed circular DNA. This is supercoil DNA. Okay, the DNA, they will automatically to form this supercoil, because remember they have to make sure the conformation will be the least free energy. All right, so that's why they become stable, right? So now, okay, let's watch this video, the DNA topology. Okay, it's very important.
It is a well-known fact that double-stranded DNA forms a double helix structure, but the double helix itself can twist and turn into different conformations. So, how does DNA change its shape, and why is this behavior important to our cellular functions? This video will use a physical model of DNA to help explain the significance of DNA topology and simulate how specific enzymes modify the conformation of double-stranded DNA. The unstrained or relaxed DNA structure has 10.4 base pairs per turn of the helix. One way to describe the structure of a DNA double helix is by a combination of its twist and writhe, the sum of which is called the linking number. The twist is described as the number of helical turns of one strand about the other, and can be measured by counting how many times one strand wraps completely around the other. For example, this relaxed linear DNA has a twist of 1, 2, 3. There is 0 writhe in this example. The writhe is described as how many times the double helix crosses over itself, and is positive or negative depending on the orientation. For example, the circular DNA has a writhe of -1 because it follows the right-hand rule and the upper strand travels left to right, and this DNA has a writhe of 1.
So remember, the right hand would be negative and left hand would be positive, right? So you can see the left hand, all right, from left to right on the top. If you go back, this is from right, right? You see the right to the left is the right hand, all right? So it would be negative, okay?
And this DNA has a writhe of 1. Both of these examples also have twists. Realistically, long DNA molecules like these can have linking numbers in the thousands. For this video, we're going to look at a physical model of circular DNA with covalently linked ends. Unlike linear DNA, this shape is topologically constrained, meaning you can't change the linking number of the DNA without breaking one or both of the strands. But what would physically happen to this DNA if you added or removed twists or writhe in the double helix? Overwound DNA has fewer than 10.4 base pairs per turn of the helix. Thus, overwound DNA is more tightly wound than relaxed DNA and has a greater number of twists and a higher linking number than relaxed DNA. When DNA is overwound and has an increased linking number, the DNA becomes positively supercoiled, and the double-stranded structure begins wrapping around itself and creating positive writhes. Regardless of these visible changes in conformation, remember that positively supercoiled DNA will have a higher linking number than relaxed DNA, and the two strands of the double-stranded DNA are more difficult to separate. Underwound DNA has greater than 10.4 base pairs per turn of the helix. Thus, underwound DNA is less tightly wound than relaxed DNA and has a fewer number of twists and a lower linking number than relaxed DNA. When DNA is underwound and has a decreased linking number, the DNA becomes negatively supercoiled, and the double-stranded structure begins wrapping around itself in the opposite direction as positive supercoiling, creating negative writhe. Regardless of these visible changes in conformation, remember that negatively supercoiled DNA will have a lower linking number than relaxed DNA, and the two strands of the double-stranded DNA can be separated more easily. But if DNA is least strained in the relaxed form, how does the double helix change from being relaxed to overwound or underwound? And how does positively or negatively supercoiled DNA return to a relaxed state? Topoisomerases are a class of enzyme that usually restores DNA to its relaxed state. This means that topoisomerases unwind overwound DNA strands, reducing the linking number back to the preferred 10.4 base pairs per turn, or rewind underwound DNA strands, increasing the linking number back to 10.4 base pairs per turn. Topoisomerases work by breaking one or two DNA strands and passing the same number of DNA strands through the break. This change results in an increase or decrease in the linking number of our topologically constrained circular DNA. There are two main categories of topoisomerase, type 1 and type 2. Type 1 topoisomerase breaks one of the two DNA strands and passes the other strand through the gap. This increases or decreases the linking number by 1. Here, the top DNA double helix represents the structure before the topoisomerase added a twist, and the bottom DNA double helix is after the enzyme completes the reaction. Type 1 topoisomerase reactions do not require additional energy. Type 2 topoisomerase breaks both of the two DNA strands and passes the entire double helix through the gap. This increases or decreases the linking number by 2. Here, the top DNA double helix represents the structure before the topoisomerase acts, and the bottom DNA double helix is after the enzyme completes the reaction. In this case, the result is that the rise of the molecule increases by 2. To make this idea clearer, this diagram shows what this change would look like if you added one positive rise at a time instead of using type 2 topoisomerase. Type 2 topoisomerase reactions require some form of energy, such as ATP or NADH. Bacteria also have a special form of type 2 topoisomerases called gyrase. Gyrase enzymes use energy to negatively supercoil DNA rather than restoring DNA to the relaxed state. Certain thermophiles also have topoisomerases that use energy to positively supercoil DNA rather than restoring DNA to the relaxed state. Because it acts oppositely to gyrase, these enzymes are called reverse gyrase. But if DNA is unstrained in its relaxed state, why would organisms have enzymes that intentionally supercoil their DNA, either positively or negatively? Thermophiles and hyperthermophiles thrive at relatively high temperatures, ranging from 45 to over 100 degrees Celsius. Positive supercoiling in thermophile DNA adds additional twists or rive to the double helix, thus increasing the linking number. These changes stabilize the structure, preventing the DNA from denaturing as easily at high temperature. Wanting stable DNA structures makes evolutionary sense, but what about the organisms with negatively supercoiled DNA? In fact, the DNA of most organisms is negatively supercoiled. Negatively supercoiled DNA provides a store of free energy that helps with cellular processes that require strand separation of the double helix, like DNA replication and transcription. Underwound DNA has a tendency to partially separate, so strand separation is easier than in a relaxed DNA structure, with more twists holding the double helix together. In short, negatively supercoiled or underwound DNA makes it easier to separate the double helix into two single strands. As you separate the double helix in negatively supercoiled DNA, you are creating more twists in the rest of the DNA, causing rewinding of the underwound strands. Rather than putting strain on the DNA topology, the DNA that is still base paired is returned to the ideal relaxed state. Plus, you have a portion of separated single DNA strands that can be used in cellular processes like replication and transcription. If you tried to separate the double helix of relaxed DNA, you would introduce more twists in the DNA and end up overwinding or positively supercoiling the double helix, which is energetically unfavorable. This video modeled how DNA topology extends beyond the double helix structure, and how topology is critical to our cell's ability to function smoothly by replicating or transcribing DNA more efficiently. Now, are you able to model?
Okay, so that's how important the DNA topology is, all right? Yeah, Mohammed.
I'm not sure if you mentioned, but is the video going to be available to watch again?
Yeah, the video be on the class recording. So you look at the later on you can look at the recording. Okay.
Sounds good. Thanks.
And then just another explanation here. So as you can see, Marina, you have a question?
Can you go back to the previous diagram? One more.
This one.
This one. So when we're talking about the linking numbers, we're physically looking at, okay, so on the first, there's an option to do a cursor for us, but I don't remember how to do it. So you can see what I'm pointing to, but
It is
first thing you have
it's called the linking number.
Where?
Yeah.
There's right.
This is what I was confused about. What units are we physically looking at?
Unit. Now
Like
there's
what-
no unit.
Not like like what sections of the DNA molecule are we physically looking at? Like to count, okay, that's one turn. This is now linking number and how it's all connected to each other. You had the diagram before. Yeah. No.
Yeah,
No, it's
this
within the video.
from a video. Let me see. Here,
No, it was earlier.
earlier.
Yeah, like right here. Like what is considered so a full
See,
360.
one turn, two turns, three turn. That's a
Okay.
twisting now. Yeah.
Okay, I thought I was looking at only half of that,
No, it's only,
and that
it,
would be considered as one.
yeah, yeah. The full turn, one full turn is one. Okay.
Okay.
Okay. And then just briefly, like this one is the what's this transcription? Yeah, it's transcription or transcription. So basically, when they go through this DNA template, then they introduce the positive supercoil, right? And then they need to have a gyrus to remove this positive supercoil. And this is the replication. So replication that this is replication. You open a double helix. When you open a double helix, then you introduce the positive supercoil before this replication form, so that you need a topoisomerase 2 to remove this tension. All right. So you have to cut the DNA and then move to the other side. So that's why the change is 2. That's how she explained in the video. Okay. And then become the negative supercoil. So the type 1 topoisomerase break single-strand DNA. Type 2 break two-strand, okay? And type 2 require ATP. Okay, require ATP so that so basically they move 2 strands, so they change 2. Each place change 2. So this, and this just to show the secondary, tertiary structure of RNA, because RNA is single-stranded, and however, when they find the complementary sequence, then they can form this structure like a stem here. You have stem and you have loop, all right? And that's the RNA secondary structures, tertiary structure, okay? So of course, if you are interested, there's a website that they several places they have this program that you can predict the RNA structure. Okay,